Hello everyone, I am Ashwini Disle, student of the Brisang Institute, batch 22. Today I am discuss on the topic of drug discovery and development. First we see the drug. Drug is any substance that is used to prevent, diagnose, treat or relieve symptoms of disease. Then drug discovery. Drug discovery is the process of identifying and characterizing molecule with the potential to safety modulate disease with the goal to bring medicine that can improve the lives of patients. Second point is drug development, the entire process of bringing a new drug or device to market. The main aim of the drug discovery is identifying a compound therapeutically useful in curing and treating disease. It involves identification of candidate, synthesis, characterization, validation, optimization, screening and assay. It takes about 12 to 15 years from discovery to the approval medicine. There are number of compounds are screened, but only one compound is approved for market use. And the next point is stages of drug discovery. There are five stages of drug discovery. First is target identification, target validation, in lead identification, lead optimization, product characterization. First is lead identification. First step is the discovery of a drug is identification of the biological origin of a disease and the potential target, that is protein, nucleic acid. There should be the efficacious, safe, meet clinical and commercial requirement. Second is the target validation. Expected molecular target, that is gene, protein and or nucleic acid is actually involved in a disease process, and that binding of a drug to the target is likely to have a curative effect. Third is a lead identification. Chemical lead is synthetically stable, feasible and drug-like molecule active in primary and secondary assay with the acceptable specificity, affinity and selectivity for the target receptor. Then lead optimization. Drug candidate is designed after an initial lead compound is identified. Pharmacokinetic and pharmacodynamic studies are an integral part of optimization. Then last is a product characterization. When any new drug molecule shows a therapeutic activity, then the molecule is characterized by its size, shape, strength, weakness, use, toxicity and biological activity. Then next point is stages of drug development. There are six stages of drug development. First is the formulation and development. Second is the preclinical research. Third is the investigational new drug. Then clinical trials, new drug application and approval. First is formulation and development. Formulation and development is the first stage of drug development. In this, physiochemical properties of pharmaceutical ingredient are characterized to produce a stable, optimal and optimal dosage form for a specific administration route. Second is a preclinical research. It involves evaluation of drug safety and efficacy in animal species. In preclinical trial can be conducted in two ways: general pharmacology and toxicology. General pharmacology deals with the pharmacokinetic and pharmacodynamic parameters of a drug, and in toxicology deals with the in vitro and in vivo study. In vitro experiments are conducted outside the animal body. In this take the animal part such as tissue, organ or plasma, and experiments are conducted in laboratory. And in vivo study, the experiments are conducted inside the animal body. In preclinical research, there are different types of animal use such as guinea pig, rat, mice, monkey, and the main aim of the preclinical research is to check the pharmacodynamic and pharmacokinetic activity of a drug, to check the safety and effectiveness of a drug, and to check the toxicological study. Third is the investigational new drug. Drug developer must file the IND application to FDA before commencement of the clinical research. Fourth stage is the clinical research conducted in people to answer specific questions that the safety and efficacy of a drug. There are four phases of a clinical trial, that is phase 1, phase 2, phase 3 and phase 4. In phase 1 there are 20 to 100 healthy volunteers are taken, and the main purpose of this phase is to check the safety and dosage of drug. The length of this phase is several months. Second is a phase 2. In this phase, the 100 to 300 patients are selected with that targeted disease, and this phase is also known as a exploratory phase. The main purpose of this phase is to check the efficacy and side effect of a drug. Length of this phase is several months to 2 years. Next is a phase 3, is also known as the confirmatory phase. There are in this phase the 300 to 3000 patients are selected with the targeted disease, and the main purpose of this phase is to check the efficacy and monitoring the adverse reaction or check the drug interaction. Length of this study is 1 to 4 years, and after the completion of phase 3 the NDA application is applied. Next is a phase 4. Phase 4 is also known as post-marketing surveillance. In this phase provide additional information after approval, including risk, benefit and use. Next is a new drug application. Express full story of drug development. Drug developer must include all about drugs starting from preclinical data to phase 3 data in the NDA. The purpose of this phase is to this NDA application is to verify that a drug is safe and effective for its proposed use in the people. After the completion of NDA application, FDA approve the complete NDA and approve the drug for market use. Thank you.